Protein structure 5. In this video, I'm going to talk about the different levels of protein structure. There are 4 of these that we will discuss. These essentially levels are like levels of organization. And they build upon each other to contribute to the protein's three-dimensional shape. So the first level is called primary. It's often abbreviated with a 1 and a little degree sign, a little dot. That's what happened today. So primary structure is simply the sequence of the amino acids in a given protein's chain. And so that might be methionine, alanine, glycine, etc. So it means which amino acids are found in what order. Then the next level of protein structure, which builds upon that sequence, is the secondary structure. And this is the formation of the initial shapes of the protein. This is when the alpha helix and beta sheet structures form. And again, that's part of the backbone. Those structures are formed due to chemical interactions between the portions of the polypeptide chain that are the backbone, not the R groups. Then, if we consider this at a kind of zooming out a little further level, then we consider tertiary structure of the protein. And this is when we consider the complete three-dimensional shape. And this, in particular, has to do with the side chains and how the different chemicals and functional groups which make up the side chains will interact with each other to produce a more complex protein structure for that particular protein. For each individual polypeptide chain, they will have primary, secondary, and tertiary folding. Sometimes we consider quaternary folding, and this is when we have 2 polypeptide chains which interact together in a stable form to make a protein complex. So we'll talk a little bit about quaternary structure now. And basically, quaternary structure can use any of the chemical interactions we've talked about for secondary or tertiary. So namely, they can use hydrogen bonds and any of the other interactions like van der Waals attractions, hydrophobic forces, etc. And I'll give you one example of a protein that you've heard about in presumably other biology courses or other courses, and that is the hemoglobin protein. So hemoglobin, as you may know, is used in oxygen transport. This particular protein is found in high quantities in red blood cells, and it is made up of 4 polypeptide chains. And this is one example for how we can talk about the different use of protein versus polypeptide. So these 4 polypeptide chains, you also may know this, there's 2 alpha chains and 2 beta chains. And only when all 4 of them fit together in their proper shape do we call this a protein, and in particular the hemoglobin protein. And here's the difference between a protein and a polypeptide chain. So basically a protein is once you've got all of the levels of protein structure put together. For a protein that has multiple polypeptide chains that fit together, then we don't call it a protein until we get to the very end. There are some other proteins that are just kind of lone wolves and they act by themselves and don't do a whole lot of complexes to be able to do their individual function. Basically, they can function just as an individual folded polypeptide chain that's folded through primary, secondary, and tertiary levels. So here's a picture, a figure to look at and see a little bit about these different levels of protein structure. So you should be able to know these examples and be able to recognize them through a figure. Basically, if we start on the upper left, then we can see the primary structure over here. In particular, this really relies on the peptide bonds between the individual amino acids. Then if we look down here, we can see just one beta pleated sheet or beta sheet, and so that is secondary structure. If we look over here on the upper right, then notice that we can pick out several alpha helices as well as some beta pleated sheets. And so that means that we're looking not just at an individual secondary structure, but we're looking at multiple secondary structures, plus some extra folding that's taking place. And so this is tertiary structure depicted here. And then last, over here on the lower right, this is actually hemoglobin right here. And so basically, we can see that there are four kind of blobs going on here, and notice that they're different colors. A lot of times the different colors will indicate that they're distinct polypeptide chains. And moreover, this particular figure has shown that there's kind of this light pink and then this like darker pink or purple. And so these are probably the two different alpha chains, and then these two are the two beta chains. They're actually identical to each other, but this is the way that this particular diagram has been illustrated. So you should be able to recognize levels of protein structure through figures and diagrams, and basically that's some hints I have for you as you're preparing for this material and learning how to apply this information to assessment questions.